Hello everyone and welcome to the video. Today we are going to talk more about species, strain, and strain improvement. First of all, if you have an environment that is suitable for bacterial growth, then you are going to get a species. Species is the basic rank representing a group of organisms that share common structural and biochemical characteristics. Strain is a subtype within that species. All strains belonging to the same species are very similar, but they may differ in one or two significant traits. So, one species can contain a laboratory strain and a pathogenic strain. These naturally occurring strains are wild strains and cannot be used in industry. They are not genetically stable and they have low production rate. So, we need to improve these strains. But what do we mean by strain improvement? We have two main categories in strain improvement: classical way and modern way. The classical way has three main processes: random mutagenesis, protoplast fusion, and adaptive laboratory evolution. The modern ways include recombinant DNA technology, metabolic engineering, and genome editing. Random mutagenesis is the most common classical method. It involves exposing microbial population to mutagens such as UV light, chemical agents, and so on. After mutagenesis, a large number of mutant colonies are screened to identify those with the desired trait. Protoplast fusion allows for the combination of genetic material between two different strains. It involves removing the cell walls of the strains to create protoplasts which are then fused. The resulting hybrid cells can combine desirable traits from both parent traits. Adaptive laboratory evolution acts like natural selection. Microbes are cultured under specific conditions, and over many generations, those that adapt and perform better are selected and propagated. We need to know that in random mutagenesis and protoplast fusion, active screening is required, which means that we need scientists to actively test and isolate the improved mutant from a large population. However, in adaptive laboratory evolution, the environment itself automatically favors and enriches the best performing strains. Both random mutagenesis and protoplast fusion are considered short-term processes. However, the adaptive laboratory evolution is considered a long-term process. Now let's move on to the modern way. Recombinant DNA technology involves directly manipulating an organism's gene. Scientists can insert, delete, or modify specific genes. Metabolic engineering is a systematic approach that uses genetic engineering to optimize the metabolic pathway of a microorganism. Genome editing is a highly precise and efficient gene editing tool that allows scientists to make targeted changes to DNA. It is a quick and accurate way to modify multiple genes simultaneously, significantly accelerating the development process. Now we develop the wild strain and get our industrial strain. Industrial strain is genetically stable, it can withstand harsh conditions, and it can produce high production rate. If you are interested to know more about microbial growth and microbial growth cycle, make sure to subscribe and watch the next video. Goodbye.